Hello and welcome to our microbiology assignment. Today's topic of presentation is the role of microbial revolution in fermented foods, the propionic acid. The group members are Ziyu, Trisha, Vinita, Ishpreet and myself, Pavitra. Fermentation is a chemical process converting carbohydrates to alcohol or organic acids using microorganisms such as yeast, mold or bacteria. Propionic acid is a naturally occurring organic acid with the chemical formula CH3CH2COOH. It plays a crucial role in various industries, particularly important in the field of food and agriculture. Propionibacteria are a group of bacteria that produce propionic acid through their metabolic processes. They are naturally found in diverse environments, including soil, dairy products, and the gastrointestinal tract of animals. They are especially known for their ability to convert lactic acid into propionic acid through fermentation. Apart from its role in cheese production, they have other significant applications. They act as a food preservative to inhibit the growth of molds and other undesirable microorganisms. The purpose of presenting this topic of propionic acid and fermented food is to understand the application and production of propionic acid and propionic bacteria in various industries. This includes the food production as new studies continues to uncover the potential benefits and explore the new avenues for their utilization. Now I shall pass it to Ishpreet to provide a detailed description of the bacteria.
Propionibacterium is a genus of bacteria belonging to Actinobacteria phylum. It comprises of several species, with Propionibacterium fruterinci and Propionibacterium acidpropionici being the most commonly studied. These microbes display exceptional versatility in food fermentation. Structurally, Propionibacterium are rod-shaped bacteria with a unique feature. They possess both gram-positive and gram-negative characteristics. Their cell walls contain peptidoglycan, which aids in cell division and gives them their distinctive shape. To ensure the successful growth of Propionibacterium, certain conditions must be met. This bacteria thrives in an environment with a pH range of 5.5 to 7.5 and a temperature of 30 to 37 degrees Celsius. They require anaerobic or microaerophilic conditions, which means they prefer low oxygen levels. Now, I shall talk about Propionibacterium and its vital role in cheese and bread production. Propionibacterium, the unsung hero of cheese production. This bacterium is responsible for the distinctive flavor and texture of Swiss and Emmental cheeses. During the fermentation process, Propionibacterium consumes lactic acid and produces carbon dioxide and propionic acid as byproducts. The carbon dioxide forms bubbles, creating the characteristic holes in the cheese, while the propionic acid contributes to its nutty flavor. And let's not forget its role in the fermentation of bread. Propionibacterium, along with yeast and lactic acid bacteria, contributes to the complex flavors and aromas of sourdough bread. It breaks down complex carbohydrates into simpler compounds, releasing organic acids that lend that distinct tangy taste that we all love. Apart from its significance in food fermentation, recent studies have suggested that Propionibacterium has potential health benefits for humans. It is believed to enhance the intestinal barrier, modulate the immune system, and even exhibit antimicrobial properties. However, further research is needed to fully understand its potential therapeutic applications. And that concludes our exploration of Propionibacteria in food fermentation. This remarkable bacteria has captivated scientists and food enthusiasts alike, showcasing the extraordinary taxonomic features, structural adaptations, optimal growth conditions, and their crucial role in creating delectable food products. Now I would like to pass the floor to my group member.
The study of propionic acid and its fermentation can be traced back to the late 19th century. In 1861, Louis Pasteur, a renowned French chemist who also made significant contribution to the field of microbiology with his discovery of pasteurization, made an observation that certain bacteria produce carbon dioxide and propionic acid during the fermentation of glucose. This bacteria were later named butyric organism due to their ability to produce both butyric acid and propionic acid. Further research in the early 20th century led to the identification of the specific microorganism responsible for propionic acid fermentation. In 1901, Orla Jensen and von Ferdinand isolated a bacterium from Swiss cheese that demonstrated the production of propionic acid. This bacterium was subsequently classified as Propionic bacterium Ferdinandi, belonging to the genus Propionic bacterium. The industrial production of propionic acid using propiogenic bacteria gained momentum in the mid-20th century. In the 1950s, large-scale fermentation utilizing propionic bacterium species were developed to produce propionic acid on a commercial scale. These bacteria were cultivated in bioreactors using substrates such as glucose or glycerol, and their metabolic activities resulted in the production of propionic acid. Over time, this industrial process has undergone continuous refinement and optimization to improve the efficiency of propionic acid production. Today, propionic acid fermentation by propiogenic bacteria remains a significant industrial process with diverse application in foods, pharmaceutics, and chemical industry. Ongoing research is focused on exploring the metabolic capabilities of this bacteria, investigating genetic modification to enhance production, and discovering new applications for propionic acid and its derivatives. The Wood-Workman cycle, also known as the succinate-propionate pathway or the decarboxylic acid cycle, is the metabolic pathway utilized by propiogenic bacteria to convert pyruvate into propionic acid. This pathway consists of a series of enzymatic reactions occurring in three main stages. In the first stage, pyruvate is decarboxylated by the enzyme pyruvate carboxylase, resulting in the formation of oxaloacetate as an intermediate. This reaction requires the presence of biotin as a cofactor. Oxaloacetate is subsequently reduced to succinate through a series of enzymatic reactions involving malate dehydrogenase and fumarate reductase. This reduction of oxaloacetate to succinate is accompanied by the generation of NADH as a cofactor. In the second stage, succinate undergoes several enzymatic reactions, including the conversion of propionate CoA transferase, propionate to propionyl CoA, and propionyl CoA to methylmalonyl CoA, resulting in the conversion of succinate to methylmalonyl CoA. The final stage of Wood-Workman cycle involves the conversion of methylmalonyl CoA to propionic acid. This conversion is facilitated by the enzymes methylmalonyl CoA acrylase and methylmalonyl CoA mutase, which converts methylmalonyl CoA to succinyl CoA. Succinyl CoA is then further transformed into propionyl CoA by the enzyme succinyl CoA synthase. Finally, propionyl CoA is converted into propionic acid by propionate kinase, releasing ATP as a byproduct.
Swiss cheese is a type of cheese that is known for its distinctive appearance and flavor. It is also known as Emmental cheese or simply Swiss. The cheese is named after the Emmental region in Switzerland where it was first originated. Swiss cheese has gained popularity worldwide and is now produced in various countries. Swiss cheese has a characteristic appearance due to the formation of holes, or commonly known as eyes, in its pale yellow interior. These holes are created by the bacteria during the fermentation process. The cheese has a firm and elastic texture with a rind that is either natural or waxed. Swiss cheese has a mild and nutty flavor with a hint of sweetness. As it ages, the flavor becomes more pronounced and complex. The texture is smooth and creamy, making it popular choice for both eating and melting. During the aging process, the cheese undergoes fermentation, which produces carbon dioxide gas. This gas gets trapped inside the cheese, forming the characteristic holes. The longer the cheese is aged, the larger and more abundant the holes become. The aging process can range from few weeks to several months, depending on the desired flavor and texture. In summary, Swiss cheese is a well-known cheese variety with a mild and nutty flavor, a distinctive appearance with holes, and a smooth texture. Propionic, a genus of bacteria, can have both beneficial and non-beneficial impacts on the human health. Let's look into the beneficial impact of Propionic on human health. Firstly, some strains of Propionic have probiotic properties supporting gut health and improving digestion. Propionic can also produce essential vitamins such as vitamin B12 or any B group vitamins, which are essential for numerous physiological processes in our body. Besides, certain strains exhibit immunomodulatory effects, stimulating the immune system and also enhancing the immune response. Propionic may possess anti-inflammatory properties, potentially reducing chronic inflammation. Lastly, they are involved in nutrient metabolism, fermenting dietary fibers, and producing beneficial short-chain fatty acids. Now let's look into the non-beneficial impact of Propionic on human health. Propionic acnes is associated with acne by triggering an inflammatory response and forming comedons, or commonly known as clogged pores. In rare cases, Propionic species can cause infections, particularly in individuals with weakened immune systems or had recently undergone medical procedures. These infections can occur in various parts of the body, including the skin, joints, heart valves, and prosthetic devices. This is due to Propionic contamination, leading to complications and delayed healing. On the other hand, Propionic can be linked to infective endocarditis, which is also known as a serious infection of the heart lining. However, it is important to remember that these impacts may vary depending on the specific strain or any individual circumstances.
Based on all the points mentioned earlier, we now have a good understanding about how the propionic bacteria works on fermented foods. So, keeping all these valuable informations in mind, now let us discuss the future prospect of this bacterium along with any potential obstacles that may arise. Considering the propionic acid, it is a naturally occurring fatty acid and a very common preservative found in everything: bread, puddings, animal feeds, and cheeses. So looking at the future prospect, these propionic bacteria are likely to continue to be utilized in the production of fermented foods for several reasons. Reason number 1. The propionic acid plays a crucial role in the production of cheese. So as the global cheese market expands, there may be increased interest in the traditional and specialty cheeses, leading to a sustained demand for propionic bacteria. Reason number 2. Exploration of new fermented foods. As the consumers continuously look out for unique and diverse food experience, they tend to incorporate propionic bacteria into novel fermented products. So for example, the plant-based alternatives like vegan cheeses or in the fermentation of other non-dairy products. Reason number 3. Functional food and probiotics. Probiotics are foods which contain live microorganisms that confer health benefits and have gained significant popularity, like Yakult and Vitagen. So these propionic bacteria, especially strains from Propionibacteria friderici, have shown potential health promoting benefits like production of vitamins and antimicrobial compounds. So these may lead to further research and development to include Propionibacterium into the functional fermented foods. The fourth reason and the final reason would be food safety. So this propionic bacteria is generally safe for consumption because of the natural microbiota of various environments. So in the context of fermentation of food, the propionic bacteria contributes to the preservation of the products through production of organic acids and antimicrobial compounds, which prevents the growth of pathogenic microorganisms. Looking over the other side of future prospect is the potential risk or challenges that could arise. So what are the challenges that are limiting the improvement of propionic bacteria in fermentations? Challenge number 1. Acidification control. So propionic bacteria produces organic acids during the fermentation process. Controlling this acidification process is very crucial for us to achieve desired flavor and texture. So on one hand, if there's excessive acid production, it could result into sourness and harsh flavors. While on the other side, if there's excessive acid production, then it would lead to inadequate preservations and inadequate textures. So, finding the right balance to control this acidification process is very, very important, and it requires a lot of monitoring and adjustments. Challenge number 2. Temperature and environmental conditions. Propionic bacteria have specific temperatures and environmental requirements for optimal growth and fermentation. So maintaining these suitable conditions such as temperature, pH and oxygen level throughout the fermentation process can be very challenging, especially in large-scale production. Deviation from these optimal conditions can affect the metabolic activity and the growth of propionic bacteria, which can result in inconsistent product quality. Challenge number 3. The final challenge would be stability and shelf life. So the shelf life and stability of fermented foods that contains propionic bacteria could actually be a great challenge. This is because the propionic bacteria could lose its viability over time, which reduces the potential health benefits it has and also the fermentation capabilities. So, ensuring sufficient viability and stability throughout the product's shelf life requires appropriate packaging, storage conditions, and formulation considerations. Addressing these challenges requires ongoing researches, technological advancements, and collaborations between researchers, food manufacturers, and regulatory authorities. So, by mitigating and understanding these challenges, the successful utilization of propionic bacteria into fermentations of food can actually be achieved while ensuring product safety, product quality, and customer satisfaction.
In conclusion, now we are aware of the history and the mechanism of propionic acid alongside the future prospects of utilizing such microorganisms in the fermented foods. Overall, the role of microbial revolution in propionic acid and propionic bacteria has revolutionized the production of fermented foods. It has not only contributed to the rich diversity of flavors and textures in the industrial culinary, but also offered potential health benefits and improved health safety using mechanisms like wood workman's cycle. As research and innovation in this field continue to progress, we can expect further advancement in fermented food production and the utilization of propionic acid in wide range of applications.